morning everyone. I welcome you all to the first session of day 2 of the conference, and the session would be based on chronic infectious diseases and will consist of one keynote address and 4 invited lectures. I will take this opportunity to welcome our chairs for the session, Professor Natarajan and Professor Lalita Gupta. Professor Natarajan is a professor in the Department of Zoology. He did his PhD from the Indian Institute of Sciences, Bangalore, and then has given his esteemed academic contribution to various institutions. He has been an assistant professor in the Department of Genetics and Plant Breeding, College of Agriculture, Dharwad. He has been a visitor Department of Ecology, Evolution and Behavior, University of Minnesota. He has been a research associate in the Evolutionary and Organismal Biology Unit, Jawaharlal Nehru Center for Advanced Scientific Research. Research scientist in Bangalore. He has been a visitor Department of Biology, University of Regensburg, Germany. Sir has mentored several PhD students and has a number of research articles and papers to his credit. Professor Lalita Gupta is a professor in the Department of Zoology, Chaudhry Bansilal University, Bhiwani, Haryana. Ma'am has done her PhD from the Banaras Hindu University, and from there on she has been a recipient of postdoctoral fellowship France, postdoctoral fellowship Colorado State University. She has been a research fellow at the National Institute of Health, USA. She has been a recipient of several Shavell Awards and a recipient of NIH Employee Special Performance and Achievement Award. I again welcome Professor Sakara and Professor Gupta to the session, and I would like them to take on this session. Thank you.
Good morning, everyone. Thank you for joining us in this session. We are already running 20 minutes late, so I wouldn't want to waste too much of time. Our first speaker is Professor KL7 Natarajan. He is a senior professor at the ACBR Center of our university. He has been working on mycobacterium tuberculosis for many years now, and has made significant contributions to understanding the disease itself. So I invite Professor Natarajan to deliver this lecture.
And it's a pleasure to be here. And at the foremost, I'd like to thank the organizers, Anju and Alok, good friends, colleagues. And it's great to be here. And I think we have put a fantastic meeting. This is not counting in my time. My count will start when I use the first one. I know Mallikarjun for a long time, so I hope Professor Lalita will also. So without much ado, let's get cracking. So we've been looking into the interactions of mycobacterium with the host system for almost 23 years. I started in 1999, soon after I came back from my postdoc, and my mentor, he promised me that he'll be here, Professor Bharat Agarwal. He's probably having jet lag, so I'll be also expecting him, and hope he comes. It'll be nice to have him here. So once I was a faculty at ICGEB, and we started the program on tuberculosis. DVT wanted us to start the program on tuberculosis. So we've come a long way, about 20-25 years, and we've identified a few molecular switches that seem to work not only in mycobacteria but other infections also. And one molecular switch that acts as a node to regulate multiple processes and regulates multiple facets of immune responses to pathogens and of course cancer. I'm just touching on one or two. Is calcium, which is the second messenger, has a very important ion. And we've identified several routes of calcium which actually play both contrasting and decisive roles. So I'll begin, and for the sake of a few people who may not be familiar with mycobacterium, is there a pointer I can use, or I use the mouse pad? So 5 bacilli are all that are required to establish a primary infection. they get gobbled up by alveolar macrophages, and the moment the system senses the infection, it actually recruits multiple cell types: dendritic cells, mast cells, macrophages, B cells, and T cells. But mycobacteria has several kits in its armory, and it avoids the immune system, and we will be talking about a few. So the pathogenesis, if I were to oversimplify it, mycobacteria, unlike other bacteria, are able to secrete many of its proteins, also called secretory proteins. And if anyone is familiar with the Montu test, which one actually goes through under the skin, it's nothing but culture filtrate. So before the sequencing of mycobacterial genome by 1996 by Stuart School Group in Pasteur Institute, everybody was working with this mycobacterial lysate and or let's say the purified proteins, using them for diagnostics, using them for as vaccine candidates, but of no use. And beyond the secretory antigens, some dead and living bacteria may also release. They are taken up by primary antigen-presenting cells or dendritic cells, go to the nearest lymph node, initiate CD4 and CD8 T cell responses. These T cells are then go back, migrate back to the infection and help macrophage kill. This is like the oversimplified way. It's true for most pathogens, but there are several caveats that occur as to what is the function of these secretory antigens. After having a smart bug, which has evolved over 2000 years, like if you visit the Koch Institute in Germany, there is a mummy that's kept there with the lungs of a TB patient, which Koch actually used that to show that this is the Koch postulate that we all talk about, the students especially. Why should a pathogen secrete proteins to stimulate the immune system? That is the question that I asked right in the beginning in about 1999. So what we did was, by that time luckily the sequence was known, and then we recombinantly expressed a few proteins. And since I was working on dendritic cells, we did one silly experiment, me and my first student. Normally what you do in dendritic cells is that you take bone marrow precursors or monocytes, culture them with GM-CSF for a few days, and then do what you want to do. In fact, stimulate, do what you do. But by mistake, I and my student added one recombinant antigen to the bone marrow cells, and lo and behold, it actually started differentiating into dendritic cells without the addition of any GM-CSF. And this was true for multiple protein CFP10, which is earlier known as MTSA or microbacterial specific tuberculosis specific antigen. ESAT6, antigen 5, and even the cell extract, sonicate mycobacteria during a sonicator and add, it will give you dendritic cells, and these are the specific dendritic cell markers, CD11C co-stimulatory markers, beautifully. But then again the question was, why should it do? And dendritic cells are one of the most important antigen-presenting cells which stimulate primary T cell responses. And immunologists can do to connect that for T cells, primary stimulatory responses, the threshold is right here. T cells are here just to prevent autoimmunity. But then we thought that, okay, we go back to the drawing board. After the release of the secretory antigen, certain dead and living bacteria may also be there. If they are taken up, that means that they will be taken up by the same dendritic cells made by the antigen in the first stage. So when we did that, we found that the immune response completely reversed. And interferon gamma, high levels of interferon gamma, if you take the antigen DCs without re-stimulation with TB, it gives you a beautiful Th1 protective response, high interferon gamma, low IL-10. But then if you do it by re-stimulating dead and living bacteria, the profile simply reverses. You have high IL-10, low interferon gamma, which means the protective response simply becomes a suppressive response. So we went on to show that it's not true for just this, and secretory of this antigen is a very complex and regulated pathway. It has most of these ESX secretion systems that the people who are working on material genetics would appreciate this a lot. And we found out that the secretion of these antigens, it has some 4,000 genes in its armory. And all the 4,000 genes, if you take this culture filtrate at different stages of its growth in the LB or the 7H11 medium, there's a specific medium for mycobacterium, you can pick and run it on a 2D page, you can pick up 4,000 distinct spots. If you do an MS, you will find out that these are all, but they are not secreted at the same time. There's a very nice regulated time-bound secretion. So we identified a few antigens. These are, let's say, within 24 hours of infection, and these are the antigens which are expressed at day 5 of the infection. This was infection of THP1 macrophages in culture, and later on we found out that if we do that in mice infected with TB or guinea pigs infected TB, we get similar profiles. These antigens 1 through 5 are not expressed at day 5, and these antigens 6 through 10 are not expressed. So they are very critical. But if you look at the function, they all induce suppression. The interferon gamma, the IL-12, the IL-6, IL-17, these are the pro-inflammatory cytokines or inducing cytokines. They are the ones that give you protection. These are all down, whereas the anti-inflammatory cytokine IL-10 is up by all these antigens, which basically means that by secreting these antigens in a regulated manner as a function of time, and then all these antigens are able to differentiate dendritic cells at the sites of infection. It creates a niche for itself for long-term persistence. See, one thing we need to understand with mycobacteria is that once you get infected with mycobacteria, you remain infected for the rest of your life. There is no sterile immunity. And unlike like Salmonella, Plasmodium, there is a time span. You will get infected for a week, 10 days, typhoid for 3 to 4 weeks. But then you get it out of the system. TB is extremely difficult to get rid. There is nothing. I'm sure all of us out here are infected, but they are healthy simply because mycobacteria has over the years evolved to infect humans. We are the only natural reservoirs. What we do with mice and guinea pigs are experimentally induced tuberculosis. Mice will also remove it if the dosage is less. So it will not kill the host, but at the same time it will not allow the host to kill. So it's a forced symbiosis that gets like if we are supposed to kill. So when we did all that, we did a lot of other stuff, but I'm coming back to my favorite molecule, calcium. We found out that by secreting these antigens, it creates a niche for itself. Any T cells responses that are mediated, they induce our suppressor. And one of my earlier students, Rajita, the first student, she also showed out that these dendritic cells act as Trojan horses for mycobacteria to migrate from, because DCs what they do is they take up antigens at sites of infection, go to the nearest lymph node, which means they migrate from the site of antigen insult. From there they go, they act as Trojan horses, and they avoids killing of mycobacteria by the dendritic cells, and they also not get processing presented on the MSC molecule, so no T cell response. But we realized that, okay, so this is like repetition. But what we found out was, in all these cases, calcium played a major role. And we found out that calcium is very important because it facilitates phagosome-lysosome fusion. You know, any phagocyte which takes up, makes a phagosome or an endosome, fuses with the lysosome, which has the lytic enzymes, which will fuse, and then for this fusion to occur, and the MSC molecules are there, so the proteins are chopped, presented on the peptides, and then you get T cell responses. So, but for this fusion to occur, two things are important. One, an intracellular concentration of calcium is very, very critical, and secondly, the pH of the phagosome must be less than 5.5. What mycobacteria does is that it actually secretes urease, which alkalines the phagosome, and also secretes ammonia, which is again like a basic in nature. So, and plus there are multiple molecules that are expressed. I will not go into the details for the short time. This is important, for this calcium is required. It also induces autophagy and apoptosis of macrophages. Calcium is actually very critical, that roots of calcium are very important. Calcium can come from various roots, from voltage-gated calcium channels, from the store-operated channels not present in the ER or the plasma reticulum, and what are known as calcium release activated channels or CRAC channels. So what we thought that when we wanted to see where is it that the calcium is coming from, we wanted to see, okay, so when we started the voltage, the CRAC channels were not like very well known. There were no inhibitors. That was a heart attack. Okay.
So my phone is on silent.
Seems mine. Thanks. So, but then there were reagents available to study voltage-gated channels, calcium channels. But then nobody had in fact worked on voltage-gated calcium channels during infections. And our report was the first to come out that way. So they were working on cardiovascular physiology, nerve physiology. Nobody thought that immune cells would also express excitable calcium channels. So we thought that, okay, why don't we do that? Let us block these voltage-gated calcium channels and see what happens. So these are the, and then these are the dendritic cells which are not expressed, which are not seen in the TV. Very bad to use the mouse. If you're in the teacher mode, you're always on the board. So these are GMCSF classical DCs. If you're infected with TB, you see
a very nice calcium spike. This is flow cytometric data. But when you take the CF15 DCs, there is absolutely nothing. So we thought that, okay, maybe calcium is coming in these DCs from the voltage gated calcium channels, so let's block it. It's like, like a water pipe, you block it here, you shouldn't see any water. And this is where the big surprise came. The moment we blocked it, we actually got further high spike in the calcium, and now we could get calcium out here. So this was weird. So I told my student to know ulta kiya. They said, right? And I said that, okay, I believe you. So we thought it's crazy, like it's like a water pipe, you switch off the tap, you should not see here, but what you see is flooding out here. So we said this is weird, and this was true even for macrophages, and this was true for L-type and R-type. There are about 6 or 7 calcium channels, R, N, E, Q type. So we found that this was the case with this, and this was true for macrophages, that was in dendritic cells, and human PBMCs. So we said, okay, so what is happening in the human samples? And then when we blocked it, we could see that they are able to get rid of the infection. So I have been talking about interferon gamma being a very important source of protection. But what mycobacteria does is that it downregulates interferon gamma receptor on the macrophage. So that even if you have a kilogram of interferon gamma on the outside, the macrophages are completely blind to it. So that's why TB patients, even if they make lots of interferon gamma, there is hardly any protection mediated by them. Okay, so we could get it in macrophages and PMC. So you see interferon gamma, because it's a downregulated receptor, even if you add exogenous interferon gamma, there's hardly any effect. So we thought that, okay, so what happens in the patients? So we looked at the expression levels of these channels in healthy controls, TB patients, and the same patients after 2 months of therapy. What happens? So you see healthy controls, this is actually my sample. I love to say this. And then there's very little expression. The moment somebody comes down with active TB, you see that the expression is just beaming, right? The expression of these channels is very high, and then the same patients, after 2 months of therapy, intensive care therapy, it comes back to normal. That means that TB pushes up the expression of these channels, locks calcium inside, and stays persistent. And we did the other, and then what we did was we infected mice with TB. Then we gave, we've done it a lot with, this is an antibody to this VGCCs. You say that it comes back. See if the lungs and this going up, down. Okay, and then we've done, see there are many L-type calcium channel blockers that are used as antihypertensives. So we did experiments with guinea pigs and all, and then we found out that we have a patent, and ICMR is now giving a trial with these calcium channel blockers as adjunct therapy to treat drug-resistant tuberculosis. So that was what it started off as the serendipity, that you say by mistake you added the CFP10 antigen in the cells, and then now we have a drug which hopefully will come out pretty soon. And we found out that when that TB, and this is I'll just quickly go through this one. Some of these stuff that it goes through the calcium and toll-like receptor pathway, the high expression of these channels, it goes through the toll-like receptor pathway. If you use siRNA to knock down the specific proteins in the TLR pathway, you see lower back expression. Okay? And then we also showed that in reverse, that if you activate the channels, what happens? So this is like amlodipine, one of the L-type calcium channel blockers, which we are using as a adjunct therapy. The trials are going on. And this is a confocal. So you see that like in H37RB, the colors are a little distorted here. This appears green on here, and this is GFP MTB, and the lysosomes is stained with lysotracker red. So the green and red will fuse in the same plane. In a confocal microscope, the color will change to yellow. If you mix it with the pen, what colors you see? So you see more phagosome lysosome fusion. So calcium, if you block it, you see more calcium. That promotes apoptosis and the phagosome lysosome fusion. So the reason why I want to bring this is that since this is a nutraceuticals conference, and then when Alok said, I said, look Alok, I don't directly work on nutraceuticals. So there's two ways of looking at it. One is that you take a few nutraceuticals and see whether it does something, and then worry about it backwards, like how is it doing it, which is absolutely fine. Then what the approach that we take is that first we work out the biology and then see whether we have a drug for it or not. So in both cases, fine as long as we get it done. So you see that novel voltage-gated calcium channels are also targets for cancer. In cancer, it seems it's the other way around. That most cancers downregulate this thing. In fact, I was wondering whether that is the reason why BCG actually works wonders for urinary bladder cancer.
Yes.
It's been working, and Anju, that it's about 30 years, it's been and prognosis is 100%. Maybe it increases anti-voltage gated channels and then it does the reverse. And we find that our favorite molecule, that's why I wanted to Agarwal here, that curcumin actually inhibits the activity of the channels. So this is something, and I wanted to refresh Bharat's memory. This was the first paper I published with him in '96. That this is a caffeic acid ferrityl ester. It's a part of honeybee comb, right? And we showed that it's a specific inhibitor of NF-kappa B, and we showed that K also actually has a direct role in voltage-gated sodium channels and the and calcium channels also, so ion channels basically. So this is where the nutraceutical component comes in my talk, that we've been looking, that we can explore, and some of my this year's fourth semester project, MSc project students, I've asked them to work on curcumin and K to look at some of the TB responses to see whether it has any influence on the regulation of potassium channels. And maybe one student from zoology this passed out this year will join me for PhD who's working on potassium channels. Calcium mein bahut kam ho gaya, bore ho gaya. So we thought that this is not true. The calcium homeostasis is not restricted to MTB. It works as a liaison with MTB HIV co-infections for a shortage of time, and Staphylococcus also. It works with Staphylococcus also, that's Staphylococcus aureus. All this is done in ACBR. All this is published, I just didn't want to put names. And also in Streptococcus. Now this is the expression of calcium homeostasis, that if you take block intracellular calcium, you see PDL1, which is an immune checkpoint inhibitor used in cancer. This gets inhibited, whereas if you use EGTA, which blocks extracellular calcium, the response is the other way. So it's just not the calcium that we have identified. The root of calcium homeostasis plays a major role. And then we just recently published a paper in cellular immunology last year, which showed that it is also restricted in involving T cell homeostasis, T cell activation, T cell function, memory to activation phenotype of the T cells, and how the effector functions of these T cells also. That if you take, let us say, calcium activated T cells, or calcium, let's say, the TMDH, intracellular block and extracellular block T cells, add those to infected macrophages, you get a reverse phenotype. You see high doses here, if you block it one channel or one calcium channel, and the T gets clear. This is all GFP BCG or GFP MTB, I forget. And here in one case it's clear. So depending on how calcium is rooted in these cells, it actually can have a devastating or a protective effect. And I think Mallikarjun, you've given me enough looks, so I will stop here. Right? So this is something that's a story. This is a project. This is a. We are also having a seminar conference in next year. Oh, sorry, next month.
Still have 5 minutes.
Oh, great. So we can take for questions. So this is the group in ICGB. Shashank was the one who identified all the voltage-gated channels. Mohit worked on Streptococcus, and the David and Deep antigens were identified by Deepthi. And this is the group in ACBR. Ankush is sitting here. He has a poster. He's looking at short-chain fatty acids in the regulation of tuberculosis, because we want to get it into non-metabolomics and the other stuff in collaboration with gut biologists who I don't see here, Rajagopal Verman. And these are the other people in the group, and I thank you all for your attention.
India, all goes through the PLR pathway.
No, that's a very good question. So there is a controversy out there. A couple of researchers showed that some of these antigens, like ESAT6 and CFP10, they bind to TLR2. It was a Nature Immunology paper, but unfortunately not many researchers could replicate those studies. So it's been taken with a pinch of salt. So that's one of the major problems. None of this, there's been this TB antigens have been worked for almost now 60, 70 years.
See, I saw several papers actually from India where they are just throwing the peptide and looking the effect.
Yeah.
How they are doing, nobody actually looked that.
But you see, we showed data show that it may not directly bind to it, but it does influence TLR responses. I will tell you, I didn't have the slides. We identified using bioinformatics, we identified an antigen called 3529C, right? So this girl, Upasana, she did that. It has a strong homology to one of the death domains in the MYD88 pathway. So, and we have another protein which has the TRAS6 domain, right?
No, see.
No, let me finish. So, when we add this, it does not bind to TLR2. But what it does is that it goes and quenches all TLR pathway. It directly interacts with IRAK1 and thereby prevents IRAK1 and MYD88 pathway. It's like a sponge. It acts like a sponge. There are many antigens which do the same thing, but they don't seem to bind to any of these receptors.
Yes.
That's
You are
like-
telling actually it goes. So here actually he is catching it out.
Yeah.
It
So
goes means how it goes. It is
it
not
goes
like
by
a,
3 receptors. Toll-like, you asked for toll-like receptors, so I said
whatever
that-
you are making like that way. It is TLR4 something I think.
No, this is an antigen which blocks TLR2 and TLR4 signaling pathway, I said. Your question was how is it going inside?
Sorry.
So there are 3 receptors that have been published. One is DC sign R1, CD209, which again one of my students is working. These are CTAG-lectin receptors. Just like your TLRs are toll-like receptors, these are CTAG-lectin receptors. These are all innate receptors. It goes to mannose receptor and also goes to MR1. The MR1 is mannose receptor 1. One more receptor, CD14. It goes to CD14. This was the earlier part. But then now they found out that the major receptor, this was a JEM paper in 2006. DC sign, which is earlier known as the GP120 binding protein because the HIV GP120 binds to it. This actually goes and binds to TB and takes it. But that it is not restricted to that. And for example, look at Plasmodium. You have antigenic diversity there also. But not a single protein of Plasmodium is shown to bind to TLR2. I'll be happy to know if you can find it. Right? So same questions come together. How does it go? Maybe it simply goes by simple phagocytosis or pinocytosis. We don't know. But the responses and effects are there. People have been working, but then.
So, Natarajan, can we also have say, can you make some of these recombinant protein, or are there any studies which suggest that these proteins are tagged with some chromosome or something? Where exactly and how exactly?
Oh, we've done that, Anju. We've done that.
Achha.
In fact, when we right after our first paper, we did that. I spent almost 6 to 7 months looking at. We knocked down TLR2, all the TLRs using siRNAs. It had no effect. So it means that then okay. We actually labeled it with PE, phytoenthrin 1 FITC, and it actually showed. By FACS, by confocal, by Z stacking, it just doesn't do. All I know is that it goes inside. And if I block it, it doesn't. If I add, let us say, some of these ligands like LPS or PAM, they don't seem to do anything to it. And I'm not the only person. The world over, this gets discussed in every team meeting. That's a question that it gets discussed, but there is no answer to this.
So another question maybe.
Sorry.
No, just I will finish that way.
Please.
So you are telling calcium channel increased by TB infection.
Yeah.
How it is doing?
Oh, I showed now where the TLR2 pathway. I skipped those slides. Yeah. So, like I said, no, it is increasing via the toll-like receptor 2 pathway. If you knock down, for example,
that's
this is
fine.
increase,
See,
and if you knock down, it goes back.
Fine, this is fine. But see how it is doing. See, your protein is
How?
expressing more or something, or which transcription factors is. See, it is TLR4,
Oh,
whatever.
you mean the transcription factor?
How it is expressing protein? You are telling receptor level that channel is increasing.
You mean the promoter activity of these channels is what you're talking about?
Either protein expression is more, or it is coming from other pool of the.
No, it is the protein expression. Oh, I see the 20 minutes is difficult to summarize
Okay.
28 papers. No, honestly, we've looked at global and microarray levels. We looked at siRNA libraries. We looked at all that papers. I have not talked about. I wanted to focus on that calcium thing, but we'll be happy to discuss it in talking.
Yes.
Last question, sir. Thank you for the nice work. Inside calcium, calcitones or internal calcium stores, they are mostly linked to the influx channels, like in T cell, as well as. So in these cases of lymphatic cell, whether did you find any correlation with this, or after using BAPTA, did you do the experiments?
Yeah. See, all the calcium channels, the calcium influx experiments are done. We use FLO4, we use ionomycin, we use some of these inhibitors that we use TMB8, EGTA. TMB8 is an IP3 receptor antagonist. It goes and binds to the IP3 receptor, thereby preventing intracellular calcium release. So that's the one. And then we use the voltage gated channel elevators and activator. Like there is a, if you it's amlodipine, which is the drug which is being like for which the clinical trials are going on. That's an anti-calcium channel blocker. K8644 is a compound which activates those channels, which actually gives you the contrasting responses. We have done those experiments.
So do you think when the people are taking blood pressure tablets, that will correlate with the TB infection?
We did that correlation there. So the TB patients, whether like any of these TB patients have like a high blood pressure or this, they think that data was, they said that we could find many people who are on these drugs and came down with TB. Okay. My mother herself was admitted to her brain tumor, but then she had a lot of lung problems also. So they said that let us try for TB. I said she will not get it because she's been taking this amlodipine for 20 years. So and it turned out it could be a correlation. But then the trials are going on. I mean, we published this data, and then Lalita Ramakrishnan, who is Venki Ramakrishnan's sister, she also works at TB. She published a paper the very next year on another drug, verapamil. So it's a combined drug trial by ICMRs. And this is part of like the drug repurposing drug we talked about. Metformin does it. Anti-diabetic drug, metformin does it. It's a huge story. But metformin is also being used. But then this is not the case. That is not that amlodipine or something. It will find a way to beat it also.
Can I?
Yes.
I compliment you. Thank you, Professor Natarajan. You've done extensive work. Some basic query which just entered my mind, not directly related to your work, maybe just my ignorance. Macrophages are very heterogeneous population. Tissue-specific variations are there. So does that impact their transformation into dendritic morphology? Tissue variation,
okay,
because
so
alveolar macrophages are the main ones which could be studied. But there are other regions where TB takes place and macrophages are.
Yeah, so you know that there is this, and we also work on tumor associated macrophages M1 versus M2, M1, M2A, M2B, M2C. So we have not looked into tissue specific macrophages, but it's a very
I just thought that maybe you are aware that somebody else,
I have not come across it. Most of it is looked with alveolar macrophages or PBMC differentiated macrophages. Most of it. Extrapulmonary TB is there, but then the theory is that it is always a migration from the primary complex into this. TB will not go directly and infect it. It has to go where the
exactly.
So
the pathway that
yeah.
you show.
So what they say is that it is the alveolar macrophages which actually take it there. Using the chemokine, that's another story I didn't say. But
And
chemokine
can the percentage of dendritic cells that are differentiating from the infected macrophages be used as markers for diagnosis? Percentage, say, a normal person may have this percentage of dendritic cells.
The window would be very low because the total population of these dendritic cells is just about 1%. So if you have 1% and then 0.1% in a population, the stats will be very difficult to see.
Thank you very much. It was excellent.
Thank you, Professor Natarajan, for a wonderful talk. I do have questions, but we can discuss them on a daily basis.
Mishra teaching.
Thank you.
Or it as a particle to all of the.
I would kindly
The
request.
routing will be from there, you know.
I would kindly request Professor Lalita Gupta to felicitate Professor K. Natarajan for such an exciting talk.